Okay, so the first process that we're going to look at is recombination. So in order to undergo recombination, you need restriction enzymes or restriction endonucleases. These are called molecular scissors because they are molecules or proteins that cut DNA at a particular point. They are naturally found in bacteria, and it's kind of like the bacteria's immune system, whereby it will cut up viral DNA because a virus wants to insert its DNA into the bacterial chromosome, and the restriction enzymes can cut up the viral DNA before it inserts, therefore it's useless really. The way that restriction enzymes cut is one of two ways, either blunt ends, which is what you can see in picture A, which is a straight cut leaving two clear ends between the DNA, or sticky ends, which you can see in figure B and C, which is where you have part of the DNA overhanging. These ones are the ones that are better used in recombination. So here is a bacterial cell that you can see up the top there. The bacterial chromosome, or the main piece of DNA, is found there, the larger piece, and there is a little plasmid on the right. It is quite small, it is a separate piece of DNA, and it does replicate separately from the DNA chromosome in the bacteria. So here is an artist's impression of a plasmid, and you can see here that there is a particular DNA sequence that is written out for you, GAA, TTC, and on the other side complementary CTT-AAG. This is called a recognition site or recognition sequence. So this is the particular part of the DNA that a restriction enzyme will cut. It's defined as a specific sequence of nucleotides, usually 4 to 6 or 8 bases in DNA, that is recognized by restriction enzymes, which then cut the DNA at that particular sequence. So you can see here the restriction enzyme that we're using is EcoR1, and it's going to cut between the G and the A and the G and the A. And because they're opposite sides, we're going to have a sticky end cut, which means that they are going to overlap slightly on each side. The way that we name restriction enzymes is by the bacteria that we find it from. So EcoR1 is found from an E. coli. So here we now have the plasmid that has been cut. So we have a space in between the plasmid that we could insert something. This plasmid is technically linear now in shape. What we then need to do is isolate a gene of interest. So a piece of DNA that codes for a particular protein that we would like to make. The beauty about DNA is that it is universal. So a bacterial DNA is just the same as DNA from a human. The types of genes that we might like to put inside a plasmid, something like the human insulin gene, or copper resistance, ampicillin resistance, which is a type of antibiotic, human growth hormone. We might like to make interferons, the proteins involved in immunity, or even we might like to make some proteins useful for vaccines. So it's really important whenever we find that gene of interest that we cut it with the same restriction enzyme that we've cut the plasmid with. That way we have the same ends, and it's much easier to insert the gene of interest into the plasmid that we have cut. So we use DNA ligase to make sure that that gene of interest has stuck into the plasmid. DNA ligase is like a molecular glue. It's an enzyme that synthesizes bonds between adjacent nucleotides. It's used naturally in DNA replication to join Okazaki fragments together on the lagging strand. Transformation is what we discuss when a bacteria has taken up this recombinant plasmid. So if you follow through the diagram here, we have a plasmid, we cut it with restriction enzymes. The same restriction enzyme would be used to cut the gene of interest out of foreign DNA. So that DNA could be another bacteria, it could be a human, it could be an animal, we don't know. Wherever that gene of interest is located, we cut it out with the same restriction enzyme. You can see here that we have some sticky ends in both the plasmid and the gene of interest. Now using ligase, we will put that gene of interest into the plasmid. We now have a recombinant plasmid. What we'll then do is add the recombinant plasmid into a fluid of bacteria and hope that that bacteria will take up the plasmid. Now that doesn't always happen, it's kind of a chance thing, but if the bacteria has taken up the plasmid, it's known as being transformed. After transformation, the bacterial cells that contain recombinant plasmids have to be isolated. So the way that we often do this is we will insert like a copper resistance or an antibiotic resistance, and we'll plate up the bacteria onto either an environment with copper, and the ones that have taken up the copper resistance and hopefully the other gene of interest that we've put in, like human insulin, will grow on that copper environment. So it's just a way of isolating which ones actually have taken up the plasmid. Then, because the plasmid replicates separately from the bacterial chromosome, that's actually undergoing gene cloning or DNA cloning, because that particular plasmid is replicating constantly, and we're going to have a large amount of that particular gene of interest. Another way to clone is through whole organism cloning, and this process is called nuclear transfer. So this is the process here. We're going to look at a cow for this particular process, but the most successful experiment that was done was Dolly the sheep, and obviously Dolly was cloned, and we had a full clone of a sheep. So the first thing we need to do is isolate somatic cells from a donor, so in this case it's a cow. A somatic cell is your everyday cell, so a skin cell, a muscle cell, a liver cell. These cells have got a diploid number of chromosomes, so both chromosomes from both mother and father. For a human, the diploid number is 46. What we need to do is take the nucleus from this cell and isolate it. We then have another donor of the same species, and we take a gamete or sex cell from the female, so that's going to be an egg. This particular cell has a haploid number of chromosomes, so half the number, and if it was a human, that would be 23. What we then do is we take the nucleus from this cell, and it is removed or destroyed, so we just have the sort of the basics of the cell without a nucleus. What we then do is we put the emptied out egg with the nucleus from the donor or the somatic cell, and through an electrical impulse we fuse the new nucleus from the somatic cell with the egg. What we then do is, with the somatic nucleus in the egg, is we culture this cell until it starts to divide. When it starts dividing, it creates a blastocyst, which is like a bag of stem cells, and we insert that into a surrogate. So in this case, it's a black and white cow. So the surrogate is now carrying the egg and is literally an alive incubator. So once that egg has developed and become the embryo and developed into a fetus, when enough time has gone past, this surrogate will give birth to a full clone of the donor who donated the somatic cell nucleus.